Hello friends, I am Dr. Vishwajit Gorpade and today we are going to talk about glycogenesis. Let us see the learning objectives of this lecture. After completion of this lecture, you will be able to define glycogen, you will be able to state the functions of glycogen, and you will be able to describe the glycogenesis pathway. Let us first see what is this glycogen. Glycogen is nothing but it is the storage form of glucose in animals. Means the glucose is usually stored in the form of glycogen in animals. It is mostly observed in liver and the muscles. The percentage of glycogen in liver is 6 to 8% and the percentage of glycogen in muscles is 1 to 2%. Although the percentage of glycogen is less in muscles, the quantity of glycogen in muscles is more as that compared to the liver. Below you can see that in muscles 250 gram of glycogen is present, but in liver 75 gram of glycogen is present. This happens because the total mass of muscles in our body is more as compared to the mass of the liver. And that's the reason why, despite of less percentage in muscles, the quantity of glycogen is more in muscles as compared to that of liver. This glycogen is present in the form of granules in the cytosol of the cells. And in the same cytosol, certain enzymes are present which are essential for synthesis of glycogen and also the breakdown of glycogen. Now let us see the functions of glycogen. The glycogen which is present in the liver, it helps to maintain the blood glucose level in between the meals. Means if we take the meal in the morning and in the evening, then in between these two meals, there is a possibility of decrease in the blood glucose level. But this doesn't happen because of this liver glycogen. It maintains the normal blood glucose level in between our meals. The glycogen which is present in the muscle, it acts as a fuel reserve to supply the ATPs during muscle contraction. Now usually fats are considered as the fuel reserve, but our body will always prefer glycogen as a fuel reserve before the fats. Why is it so? Let us see. The first reason that the glycogen is preferred as fuel reserve in our body is its rapid mobility. As it is rapidly mobile within our body, it is easily accessible. The second reason is that glycogen can generate energy even in absence of oxygen. Means it can generate energy in anaerobic conditions. The third reason is that glycogen is capable of continuously supplying the glucose to the organs which regularly require glucose, such as brain. If we consider fats, they will have certain drawbacks. Means the fats, they do not show rapid mobility. Fats, they do not generate energy in absence of oxygen. They always require oxygen to generate energy. And thirdly, fats are not capable to continuously supply the glucose. This is the reason why our body prefers glycogen as the fuel reserve before the fats. Now let us see what is glycogenesis. Glycogenesis means synthesis of glycogen from glucose. Usually this glycogenesis occurs in cytosol and it depends upon ATP, UTP and glucose. Now what is this UTP? UTP is nothing but it is uridine triphosphate. This uridine triphosphate, it is made up of uracil base which is attached to the ribose sugar, and this ribose sugar is further attached to 3 phosphate groups. The first phosphate group will be alpha phosphate group, second one will be beta phosphate group, and third one will be gamma phosphate group. Now let us see the glycogenesis pathway. The step 1 in this glycogenesis pathway is synthesis of UDP-Glucose, that is uridine diphosphate glucose. For that, initially glucose is converted into glucose 6-phosphate in presence of enzyme glucokinase. Here, phosphorylation of glucose takes place due to ATP, where ATP will provide one phosphate group to the glucose and there will be formation of glucose 6-phosphate. Now this step is the first step of our glycolysis pathway. After that, this glucose 6-phosphate is going to get converted into glucose 1-phosphate, and this conversion will happen in presence of enzyme phosphoglucomutase. Now, this is a mutase enzyme, and as we all know, we have studied it in our glycolysis lecture, that mutase enzyme helps to transfer certain group from one position to another position in a molecule. So similarly, here phosphoglucomutase will transfer phosphate group which is attached to the C6 carbon of the glucose to C1 carbon of the glucose. Here, phosphoglucomutase will first add its own phosphate group to the C1 position of this glucose 6-phosphate, and then it will remove the phosphate group which is attached to the C6 position of this glucose 6-phosphate. As a result, finally, there will be formation of glucose 1-phosphate. Keep one thing in mind, this phosphoglucomutase doesn't simply remove the group from C6 position of glucose and attaches it to the C1 position. Phosphoglucomutase will first add its own phosphate group. It is already containing one phosphate group, which it will add to the C1 position of the glucose 6-phosphate, so that there will be formation of glucose 1,6-biphosphate intermediate, and thereafter the same enzyme phosphoglucomutase will remove the phosphate group from the C6 position. As a result, we will get glucose 1-phosphate. Now, once glucose 1-phosphate is formed, this glucose 1-phosphate will interact with UTP in presence of enzyme UDP-Glucose pyrophosphorylase to form UDP-Glucose. Now, in this interaction, the pyrophosphate group from this UTP will be removed, and uridine monophosphate which is formed will be attached to this glucose 1-phosphate to form uridine diphosphate glucose. Now, how does this interaction takes place? Let us see. So, towards left hand side, you can observe the sugar phosphate molecule, and towards right hand side, you can observe nucleoside triphosphate molecule. This sugar phosphate, it resembles our glucose 1-phosphate, and this nucleoside triphosphate, it resembles our uridine triphosphate. Here, the oxygen of the phosphate group attached to the sugar is going to act as a nucleophile, and it will attack the alpha phosphate group of nucleoside triphosphate. As a result, the bond in between this alpha phosphate group and this oxygen is going to break, and the pyrophosphate will get removed from here. Once the pyrophosphate is removed, this sugar phosphate will go and get attached to this nucleoside monophosphate, which will remain after removing the pyrophosphate. So let us see how this interaction happens. So the bond in between the alpha phosphate and that oxygen is going to break. The pyrophosphate will be formed, which will be removed, and now the sugar phosphate will get attached to nucleotide monophosphate to form sugar nucleotide, that is nucleotide diphosphate sugar. Now this nucleotide diphosphate sugar over here is nothing but uridine diphosphate glucose in our case, because the sugar in our case is nothing but it is the glucose, and the base in our case is nothing but it is uracil. So the UDP glucose which will be formed in this case will appear like this. So here you can see that UDP glucose consists of uracil base which is attached to the ribose sugar. This ribose sugar is attached to the phosphate groups, and the end phosphate group is further attached to the glucose. Now in step 2, there will be formation of glycogen primer. Now in this formation of glycogen primer, the protein glycogenin is involved. What will happen over here is that the hydroxyl group which belongs to the tyrosine of this glycogenin protein will attack the C1 carbon of the glucose of this UDP glucose. Now when this hydroxyl group will attack the C1 carbon of glucose of this UDP-glucose, the bond in between this UDP and glucose will break, and that glucose will go and get attached to this hydroxyl oxygen. As a result, the UDP will be released and there will be formation of glucosylated tyrosine over here. Now this process happens in presence of enzyme glucosyl transferase. So this glucosyl transferase catalyzes the formation of glucosylated tyrosine. Now once the glucosylated tyrosine is formed over here, the second UDP glucose molecule will come, and this second UDP glucose molecule will be attacked by the hydroxyl group which is attached to the C4 position of this glucose molecule which is already attached to this glycogen. Now the hydroxyl group of this already attached glucose molecule at C4 position will attack the C1 carbon of this second UDP glucose molecule, and again there will be bond breaking. Once the bond breaks, the second glucose molecule will come and it will get attached to the first glucose molecule which was already attached to the glycogen. Now this process continues further until there is formation of chain of 8 glucose molecules, and this entire structure which consists of glycogen attached to the 8 glucose molecule is known as glycogen primer. Let us understand this process through animation. So here towards right hand side you can see the glycogen. This hydroxyl group, it belongs to the tyrosine of this glycogen protein. Towards left hand side you can see the UDP glucose molecule. Now this hydroxyl group of tyrosine, it is going to attack the C1 carbon of this glucose. Let us see how. So here I have given the numbering to the carbons of this glucose. This is the C1 carbon, this is C2, C3, C4, C5, and the CH2 carbon is the C6 carbon. So now this hydroxyl group is going to attack this C1 carbon, due to which there will be breaking of bond in between C1 carbon and UDP. Also there will be breaking of bond in between oxygen and hydrogen, and then this glucose will come and get attached to this oxygen. It will happen in this way. So the bond in between UDP and C1 carbon has broken in case of UDP glucose. Also the bond in between oxygen and hydrogen has broken. There will be formation of H plus ion and UDP. They will go away and the glucose molecule will link to the oxygen of tyrosine. So here is the formation of glucosylated tyrosine. Now second UDP glucose molecule will come and its C1 carbon will be attacked by the hydroxyl which is present at the C4 position of this first glucose molecule which is already attached to the tyrosine. The interaction will be same. There will be breaking of UDP and glucose bond. There will be also breaking of bond between oxygen and hydrogen. UDP and H plus will get removed, and now there is formation of a glycosidic linkage in between first glucose and second glucose. So here this is the first glucose, this is the second glucose, and in between there is formation of glycosidic linkage. We call this glycosidic linkage as alpha 1,4 glycosidic linkage. Now we are calling it as alpha 1,4 glycosidic linkage because here there is linkage in between C1 carbon of second glucose molecule and C4 carbon of first glucose molecule. Now why we are calling it as a alpha? This is because the glucose molecules involved in this chain, all glucose molecules will be alpha glucose molecules. Now what are these alpha glucose molecules and beta glucose molecules? Let us see. So here you can see the images of alpha and beta glucose towards the left hand side. In case of alpha glucose, the hydroxyl group which is attached to the C1 carbon, it is always present below the ring. So if we consider that this glucose ring is a plane, then this hydroxyl group which is attached to the C1 carbon of this glucose will be present below the plane. Whereas in case of beta glucose, the same hydroxyl group will be present above the ring, means it will be present above the plane. In our case, all the glucose molecules which will be attached to this glycogenin, all of them are alpha glucose molecules, and that is why this linkage is known as alpha 1,4 glycosidic linkage. Now thereafter, the number of glucose molecules will increase. It will go to 3, 4, 5, 6, 7, and finally 8 glucose molecule will get attached over here, and this is nothing but our glycogen primer. So our glycogen primer, it consists of glycogenin, which is further attached to 8 glucose molecules, means there are 8 glucose residues over here. Now we are calling it as a primer because primer is nothing but it is a base on which the construction of something big takes place. Now here, glycogen primer will act as a base for the formation of a complete glycogen polymeric molecule. So we are going to call it as a glycogen primer. Now once the glycogen primer is formed, step number 3 will take place, that is chain elongation by glycogen synthase enzyme. Now here you can see the glycogen primer which was formed in our previous step. In presence of enzyme glycogen synthase, 13 more UDP-Glucose molecules will come here, and there will be formation of a long glycogen chain over here. Now you can see over here, this glycogen primer, which was initially containing only 8 glucose molecules, has got converted into a glycogen chain which is now containing 21 glucose molecules. You can see here, initially there were 8 glucose molecules, 13 more glucose molecules have been added by these 13 UDP glucose molecules. As a result, 8 plus 13 glucose molecules is nothing but it is 21 glucose molecules. So 21 glucose molecules are now attached to the glycogenin, and we will call it as a glycogen chain. In step 4, there will be formation of branches in glycogen. This is because if a long chain of glycogen is formed, and if the chain length is still increasing because of this glycogen synthase enzyme, then there will be decrease in the solubility of glycogen. In order to increase the solubility of this glycogen, the branching is must. Also, the branching can increase the number of non-reducing ends in glycogen. What are these non-reducing ends? We will see afterwards. But here, let us see how the branching happens. So here you can see the long chain of glycogen which is containing of 21 glucose residues. In presence of enzyme amylo 1,4-2,1,6 transglycosylase, there will be formation of branch. Now how this branch is forming? Initially, the glycosidic linkage in between this 10th glucose and 11th glucose is going to break. And then this 11th glucose along with the remaining glucose molecules is going to get attached to the 7th glucose molecule over here, and it is going to get attached by alpha 1,6 bonding. So the process is simple. The bond in between 10th glucose and 11th glucose will break. The fragment of glucose molecules from 11 number glucose to 21 number glucose will go and get attached to the 7th number glucose over here. So you will get a branched glycogen chain over here. Let us understand this process from the animation. So here you can see a long glucose chain which belongs to the glycogen. Here the glucose molecule which you are observing towards the left hand side, it will form the non-reducing end of this glycogen chain. Now what is this non-reducing end? Let us see. We will understand this non-reducing end concept from the example of maltose. Now in this maltose, there are 2 glucose residues or 2 glucose units which are attached by glycosidic linkage. Now the glucose molecule which is present towards right hand side, the C1 carbon of that glucose molecule is completely free. Means it is not forming any glycosidic linkage with any other glucose molecule. So it is completely free. And as it is completely free, it can form carbonyl group upon opening of this glucose ring. Due to this reason, we call this end as the reducing end. On the other hand, the glucose molecule which is present towards the left hand side, the C1 carbon of that glucose molecule is forming a glycosidic linkage over here, means it is involved in a glycosidic linkage. This indicates that this C1 carbon of the glucose molecule present towards the left hand side is not free. It is involved in the glycosidic linkage, and that is why it cannot form a carbonyl group upon ring opening, and hence this end will be known as non-reducing end. Now let us give the numbering to the glucose molecules in this chain. Let us say that this molecule is the 7th number glucose molecule. Thereafter will come 8th number glucose molecule over here. I have shown them by dots because I cannot draw more glucose molecules in this canvas. So here will be glucose molecule number 8, here it will be glucose molecule number 9, then glucose molecule number 10. This oxygen over here, it belongs to the hydroxyl group of glucose molecule number 10 over here, and this oxygen is further attached to glucose molecule number 11, and the glucose molecule which forms the non-reducing end, it is the glucose molecule number 21. Now when the bond breaking will happen, it will happen over here, means in between this oxygen which belongs to the C4 carbon of the 10th glucose and the C1 carbon of the 11th glucose. Once this bond breaks, the C1 carbon of 11th glucose will get attached to the C6 hydroxyl oxygen of this 7th glucose. Let us see how does it takes place. So firstly the bond will break, and once the bond breaks, the hydrogen will go and get attached to this oxygen which belongs to the C4 carbon of 10th glucose, so that there will be formation of hydroxyl group, and now this 11th glucose along with the remaining fragment will go near the C6 hydroxyl group of 7th glucose molecule. Now the bond in between this oxygen and hydrogen of this hydroxyl group will break, and there will be formation of bond in between the C6 oxygen and C1 carbon of 11th glucose. This is how branching takes place. Now here in this branch there is formation of alpha 1,6 linkage. Keep one thing in mind, this alpha 1,6 linkage is not a glycosidic linkage as that of alpha 1,4. Now here we are calling it as a alpha 1,6 linkage because it is a link formed between C1 carbon of 11th glucose and C6 carbon of 7th glucose. Now here there will be formation of two non-reducing ends. Initially there was only one non-reducing end in the long glycogen chain, but now due to branching there is formation of two non-reducing ends. First end is the original end of our previous long chain, and second end is the newly generated end where the bond has broken by the enzyme transglycosylase. Now in step 5 there will be formation of granular glycogen. Now here you can see the branched glycogen which was formed in our previous step. Now there will be again chain elongation and chain branching which will occur in this already branched glycogen. Means chain elongation will again happen at these two branches, and once the chain elongation happens, again there will be branching in these elongated chains, and this will happen in presence of enzyme glycogen synthase and amylotransglycosylase. So if already formed branches undergo elongation and further branching, finally we will get glycogen molecule which will look somewhat like this. Now you can see the image of glycogen which is shown towards left hand side. See, this is the glycogenin to which initially glucose molecules got attached to form a long chain. To this glucose molecule which is orange in color, purple colored branches were formed. Now to this purple color branches, again further branching has occurred. Means elongation and branching is a continuous process over here to form glycogen at the end. The overall reaction for addition of each glucose residue in this glycogenesis has been shown over here. So there were n glucose residues. One glucose residue gets added to these n number of glucose residues in presence of 2 ATPs to form glucose n plus 1 molecules in the entire glycogen chain. There will be formation of 2 ADPs and 1 pyrophosphate group. Now in this process, 2 ATPs are required because 1 ATP is already required in the step number 1, where glucose is phosphorylated to form glucose 6-phosphate. And second ATP, it is required for the conversion of UDP into UTP, because each time whenever there is breaking of bond in between UDP and C1 carbon of glucose, that UDP should be converted into UTP, and that UDP is converted into UTP by the ATPs, and that is why 2 ATPs are required in this process. So these are the references which I have used for the preparation of this video lecture. Thank you. In our next lecture, we will see glycogenolysis.